Okay, so first one, I'm going to do prophase. That's what we have first. I'm drawing mitosis, so prophase. So what's happening, you have late and early, and again, the names come from which one is, when they're looking at it, basically. So what's happening during prophase? So you have your chromosomes looking like X, and there is the nuclear envelope that starts to break. So this starts to break, and then you also have something called, they look like churros, I would say. They look like churros. These are centrioles, and these starts to make protein fibers that are going to basically catch onto the chromosomes. So they make these protein fibers. So this is prophase. This is mitosis. And then it's gonna go into metaphase. Can anybody remember what happens in metaphase?
They lined up in the middle.
The line in the middle. So these axes align perfectly in the middle. I'm just gonna try to draw those centrioles. And these protein fibers are pushing and pulling on these chromosomes, and they line up in the middle. So that's the metaphase. And again, you can remember M as middle. They line up in the middle. And then next one is, because they're getting pulled so hard on these, they're going to break in half. So these axes break in half, and that's anaphase. And they start moving away. So A for away, maybe, from each other. So they start looking like these V's.
Yeah, they started pulling off from each other.
Yes, they start pulling away from each other. That's anaphase. And then the last one I said was telophase, and that's the one that's different between animal and plant cell. So during telophase, nuclear envelope starts to form. So you have these chromosomes, and then the nuclear envelope, this is nuclear envelope, starts to reappear. Same thing here, because it went through cell division, now you have two of those. And same thing for the plant cell. And you're gonna see the plant cells have this boxy shape because of the cell wall. And same thing happens, nuclear envelope starts to reappear. But one thing that's different is that this animal cell has this dip here, and then the plant cell has this cell plate in the middle. So it has a cell plate. So this is animal cell, and this is plant. And this is called cell plate. And then this is cleavage furrow. To cleave means to cut. So you cleave something. Furrow is basically this dip. When you furrow your eyebrows, it's gonna give you, a little bit of a dip like that. So it's called cleavage furrow. So are there any questions on this? No? Yes? Truth? I can only see Truth. I can't see anybody else. Yes, Nancy, what's your question? Yeah, I can zoom out. There. So let's go back to the images I showed you guys. So here, if you read the definitions, it's gonna say chromosomes are condensed and they become visible because when they're condensed, you can see them. Spindles form. Spindle fibers are the protein fibers they act like the spider webs. They pull on these chromosomes and they line up in the middle. Let's say they pull until they line up in the middle. That's metaphase. And because they're pulling on them, they split in half and they look like these letter V's. And now you have the information that's split in half. During telophase, the nuclear envelope starts to reappear on both of them. And for the animal cell, this is the cleavage furrow that happens. And for plant cell, the cell plate appears. And then the cytokinesis, cells start moving away from each other. Cyto means cell, kinesis movement, and they split into two daughter cells. Then each of these daughter cells starts to go through G1 phase, then S phase, then G2 phase, and then mitosis. So let me show you what you guys would have seen if we were meeting in the lab today. So what we would have done, I would give you slides and then have you find each of these. So here, some of them are labeled, some of them are not labeled. The slides I would have given you would not be labeled. And then you would look at it. And usually the metaphase is the easiest one to see when it's in the middle. And then you would go through these slides and say, oh, this is where the metaphase is happening. This is where the anaphase is happening. So here, this one is labeled. So metaphase, this in the middle, it's easy to see. Anaphase, they're kind of away from each other. Telophase is starting to divide. So this is what you would have done. Here's a good anaphase here. This is what you would have done in the lab today. So I think it's very harmful to your education that you guys are not able to see these things. And you learn it a little bit better too. Another slide we would have looked at is the fish egg cells. That's the animal. So the onion root tip is the plant. So this is what that would have looked like. So it's a little bit harder to see. It's a little more blurry. So this is interphase, and it's growing. Prophase is getting ready to be lined up in the middle. Metaphase in the middle. Anaphase, they start pulling away. They break in half. And then early telophase, sometimes some books label it early telophase when it's still kind of pulling, and then late telophase when the nuclear envelope is there. So that's another thing that you would have seen in the lab today. Any questions before I move on from mitosis? I'm going to ask you a question. Is cytokinesis part of mitosis? No. What about interphase? Is that part of mitosis? If not, when interphase happens? So no. When does interphase take place? Before mitosis. What are 3 parts of interphase? So what are 3 stages of interphase? G1, S, and G2. G1 is a growth phase, so the daughter cells grow. S stands for synthesis, DNA replication happens. G2, it grows a little bit more before mitosis. So very good, you guys are following me. This is the slide that's showing you the animal cells. So first sign of cytokinesis is this cleavage furrow, so that dip that happens. And once it's divided, the cytokinesis is complete. Now you have 2 cells. So you have 2 cells that has the DNA information or the genetic information. So what's happening during these different phases? There are checkpoints. So there are checkpoints. So just to make sure that everything is happening in the correct order, the correct bases are being added or nucleotides are being added, but every now and then your body makes a mistake and it loses the control of cell division. So that's cancer. Cancer is when the cells, they start growing out of control. They just divide and divide and divide because something went wrong during cell division and that checkpoint was not able to figure that out. So that's cancer. And there are two types. I don't know if maybe you guys probably heard, oh, if it's a benign tumor, it's not dangerous. That's not true. This also means, there is something wrong. So maybe if you get a really bad sunburn or some sunspots, and then you go to your doctor, and you get checked out, and they just say, oh, it's a benign tumor, there maybe there's still something that needs to be taken care of. So a tumor is abnormal mass of tissue. The cells of tumor divide out of control. They divide and divide and divide. They can be benign or malignant. So what's the difference between these two? Benign, it doesn't mean that it's not dangerous. It just means it's not spreading. It's just in one place. So the cells are dividing uncontrollably just in one place. Um, Whereas this one here, well, you can see what's happening here. So the cells were dividing, and what started to happen, some of those cells started to leave, and it went to a new location and started to divide there. So it happens in multiple places. Basically, these tumor cells are able to travel to different parts of body or metastasize, and then the new tumor starts to grow there, and new tumor starts to grow there. So it's very dangerous. So lymphatic vessel is a vessel in your body that has, that's basically responsible for your immune response, but then it's not able to basically control this sometimes. So that's two types of tumors. So nowadays what they can do, they can scan for some of the genes that can cause cancer. They can see if you have that gene or that mutation that can cause cancer. And the one very common one is a BRCA gene. Okay. So this is the one that that's known. If you have this gene, when they scan your genome basically, and if this gene lights up, means you have this gene, then they said there is 100% that you're going to get cancer. And some famous people, Angelina Jolie. So so her mother and sister, they had cancer. So they found that they had cancer, and then she did the test, and then it lit up. So basically, she had that gene, so BRCA gene, and before she even got cancer, she got basically proactive, and she got rid of her breast, and also I think she got removed. She got her ovaries removed. So that was even before she got any type of sign that she's going to get cancer. But she listened to the doctors, and because it was in her genetic history, she just went ahead and she did the preventative care for that disease before she even got the disease. So this is what they scan for now. And also another thing I want to mention is, some of the factors that can cause cancer. So if it's a family history, if it's genetic, then it cannot be avoided, unless you do something like Angelina Jolie did. You just get rid of the part that might become cancerous in the future. Another thing that really matters is the detection time. So she did it before, she did the preventative method before she even, had cancer. Whereas nowadays, insurance companies, they don't really cover for any of the preventative methods. They don't want to pay for anything unless you really have the disease. There are some cancer-causing viruses. Human papilloma virus is one of them, and it can cause cervical cancer. So maybe if someone gets the virus, they can get cervical cancer. UV radiation, I mentioned before too, UV radiation, so try to wear sunscreen. And then tobacco use. Also, the vapors some people use, they just haven't gotten around to basically limiting that. So that can weaken your immune system as well. And then unhealthy diet. One thing that's not on this list, but it's well known, is alcohol abuse. So that can also weaken your immune system, and it can basically mess up your um, genes and cause mutations. And so there are different things that can cause cancer, and you can't really avoid it sometimes. And then it also depends. So genes and the environment, both, they can increase cancer risk. So it's not just one thing, it can be multiple of these factors. So again, cancer is when the cells are growing out of control. Some of the cancer treatments, they include surgery, so complete removal, what Angelina Jolie did, chemotherapy, so chemicals, and then radiation. So people can go through these procedures and they can basically take care of the tumor. Um, Sometimes it also depends, on your age. Basically, let's say you're 80 or 90 years old, sometimes person just wants to, not go through these procedures and just keep their dignity. They had a happy life, they just don't want to go through any of this. So sometimes the cure is worse than the disease, and it's not always worth it. So some people just decide not to do it. Another thing I want to kind of mention is that the humans, they've been been around for a long time, millions of years. But before the modern medicine came around, the average lifespan was 30, 35 years, maybe 40 years old. And then, if you talk about evolution, the purpose of the species is to reproduce. And by that age, the species were able to reproduce, and there was no purpose of sticking around. So the body starts to kind of break down after, 35, 40, 50 maybe. But nowadays, the lifespan has increased like crazy. And that's because of the modern medicine. So if you go to maybe your grandparents' house, you're going to see their pills are lying around almost everywhere. And that's to basically prevent breakdown of the body. But our bodies naturally are not built to live that long. But with the use of drugs, we are kind of getting around that, which is really good. So again, treatments can include different types: drugs, chemotherapy, and radiation. I think that's it. That's all I have for you guys. So are there any questions?
Hi, Professor. I was having internet problems this morning, and I missed